What are the main drivers of evolution in nature? If you think in the usual thinking of Darwin's natural selection, you will immediately come with the idea that competition and the survival of the fittest is what is relevant. When Darwin formulated his theory, actually, he included two important pieces. One was the observation that nature is diverse and that somehow, at that time it was not possible to know, somehow there's a variability, there's a diversity that is there, that appears in a natural way. And from that range of possibilities, there's a selection process. And the idea of selection came to Darwin after reading a very important essay by Malthus, who actually suggested the idea that under limited resources, not everyone can survive. And by connecting the two parts, natural selection theory came about. And from that picture, we tend to think that survival of the fittest, so who is the strongest in some way, is what actually drives everything in evolution, and maybe in ecology. In this lecture, we will discuss how to formulate the simplest mathematical theory of competition, which is going to be one particularly relevant piece of our mathematical frameworks. But also, we'll discuss a very important part of the story, probably more important in many ways, which is that beyond competition, there's cooperation. And cooperation has been a major driver of evolution, in particular in driving new novelties. So how we can formulate a theory of competition? We are not going yet to talk about evolution, because mutation is something we will discuss in a later lecture. But an important part of what we need has to do with the following idea, which is very simple to formulate, right? As with everything else, we'll restrict ourselves to one dimension. But the idea is that assume that we have the simplest case. We have two species with populations x1 and x2 are indicated here. Each species is able to replicate with given rates, and they share some resources. Resources here can be available space, or can be, more commonly, metabolites that they want to use from the environment. This is a limited resource, and so because of that, not everybody can survive. And the two species will try to exploit resources the way they can, and the big question is, what's the impact of that dynamics? Okay? So that requires formulating a kind of mathematical model that we will use extensively, which introduces a very restrictive rule, which is that populations are constant. Meaning that the total amount of individuals that are at any time in our given case study remains the same, right? So x1 and x2 might change, but the overall amount of individuals here, bacteria or whatever it is, it will remain constant. Usually this is formulated as the constant population constraint, because we are going to impose that, right? That implies that some of the things we are going to derive from the mathematics might change when this constraint is not there, but it's an extremely powerful tool because it allows to simplify the mathematics and allows to reach very well-defined conclusions. So let's consider the following scenario, right? Imagine that these x1 and x2 are cellular populations, right? One population, for example, is one kind of cell type, let's say normal tissue that replicates at a given rate, right, under given conditions. And let's assume that there's another population, right, which is going to be cancer cells. They also replicate with a different rate. Okay? We will have time to discuss about cancer in different contexts, but it's an interesting problem that can be actually manipulated under laboratory conditions, right? We can actually define this problem that we are going to describe formally in an experimental way. And suppose that we want to make a model that describes the change in time of normal cells and cancer cells, okay? And we know they compete. They compete. Imagine, for example, we are making an abstraction here that they share, for example, a given space. And they're trying to compete. Cancer cells might want to invade the tissue that is occupied by normal cells, right? Somehow, these two populations, right, interact and compete in trying to exploit resources. All right, so if I want to formulate my model, I need, in principle, two differential equations. One is for the speed of change for normal cells, and the other for cancer cells, right? One thing that we have in mind is that whatever it comes out from here, if I analyze this problem, I should end up in one particular case, which is if I only have one population, this should be a logistic problem, right? I grow with limited resources until you have a limit of the total population. So how can I formulate that? Well, one clear thing is that normal cells replicate proportionally to their populations. That's standard, we know that. Cancer cells will also replicate proportional to the populations. I'm not assuming a number of important things here, like, for example, cancer cells more typically will come from cancer stem cells, so it's a tissue architecture that we are not considering, but we will. And that would be for the replication part. If I don't do anything else, this is going to be exponential growth, right? But that cannot be. Somehow, right, I have to have an inhibition from the effects that I don't know yet how to formulate. There's some kind of interference between populations. But what I can say is that wherever it is, the inhibition term, right, should be proportional to the number of cells in each compartment. Each cell in each compartment feels some kind of inhibition. What's the form of this function? I don't know. Let's write down this in this way, okay? It's something that I need to find out, right? It's totally general. So let's use that, and now I'm going to use this constraint, right? I make the assumption that the total population is constant. So we know that. It means that I reduce the dimensionality of the problem, because what I'm saying is n plus c, okay, is a constant, okay? For simplicity, because it's convenient for us, we can say that n and c are going to be the fraction of cells in each compartment, right? So this is normalized, and I can say that the total sum is 1, okay? If that's the case, then it's no big problem to see that my two-dimensional system collapses in one dimension, right? If I know C, I know N. All right, so phi is what we don't know, but it's easy to find. If I assume this constraint, all right, an immediate consequence of that is that because this is constant, this derivative is 0. The derivative of a constant is 0, which means that the sum of derivatives is 0. And armed with that, we can do something very nice. If that's the case, these two equations here, right, if I make the sum, has to be 0, which means that this part and this part, okay, which means these, right, have to be 0, okay? But by construction, we said, let's assume that this is the constant, this is fractions, so this part here is 1. And as you can see, we immediately find out who is phi. The function that introduces the competition term, actually, is nothing but this. Okay? So we know what it is. So this has a number of consequences. The first one is that I can reduce my system into one dimension and explicitly calculate, for example, for cancer cells, okay, how I can get into the one-dimensional system. I have this term here, minus C, and now I know phi, right? Phi is just this here. But since n is 1 minus C, okay, I can get rid of it. And I already have here an equation that collapses everything into a single variable. Let's make it nicer. If you look at this carefully, we see that we have RC by C here and here, right? So I can take some common terms, have this multiplied by 1, and this multiplied by minus C. Okay? I'm almost done because the only thing that remains here is almost the same except for this factor here, right? So I'm there. The time evolution of my cancer cell compartment is going to be a parameter, right, which includes the two replication rates, which essentially weights what's the difference between them, right? This is a logistic equation, not different from what we know, except that the parameter here is the difference between the replication rate of cancer cells and replication rate of normal cells. But the outcome is extremely clear. We can make a stability analysis and whatever you want. Clearly, as with the logistic system, we have two fixed points. 0, the extinction of the cancer cell population, there's no cancer cells, or 1, which means that you are completely occupying all the space. Okay? And it's immediate to know who's stable and who's not, because if you look at this difference, if the cancer cells replicate faster than the normal cells, what you have is a positive constant here, and you will have just logistic growth, right? Growing up until you complete this attractor here. That will be stable. So I'm going to observe the whole population of cancer cells occupying everything if its replication rate is larger than the replication rate of normal tissue, right? And the other way around for the other. The extinction attractor for cancer cells is going to be stable if they are not able to overcome the fastest replication normal cells, right? So you see, this is a simple, very clean way of actually exploiting not only this particular scenario, right, but any scenario where the two populations they have somehow compete between with each other, right? And that will be, in a general, something we can apply in other contexts, from simple models of growth to game theory, right? And we will see what are the consequences of that. Just as a little comment about that, what I just defined here can be generalized for many dimensions, and sometimes it's something that we can, again, collapse into one dimension. But just to tell you, if I want to define this problem in general terms, okay, for each population, I will have a growth term minus something now that might depend on many populations, right? So my vector here just reflects that they have S species. So this is a vector in real space and dimensions. I could do exactly the same approach, and you can easily show that your phi function, right, is just the sum of all the replication rates multiplied by this, right? This is again under the constraint that I just mentioned, that this is normalized, right? And that brings into something that I just wanted to briefly mention, that when you do that, what you actually do is define this in terms of how fast I am in replicating compared with the average population behavior. Why is that? Because if you take this equation here, all right, what I'm saying here is that the constant population constraint, right, in this general framework that makes me able to define this phi function, right? Let's write it here. It can be interpreted in statistical terms, because if I have into account that these x are normalized, so they are probabilities, right? In reality, this here is the average rate of replication, right? In other words, if I write down this statistically, this is the mean value, right? On average, how well the population replicates. And that's very nice, because that means that I can write this, and you will see the implications of that at some point. No, that's correct. What I do actually when I write these equations, and that's, as I was saying at the beginning, in a way is the first component of Darwin's theory of natural selection. What I do is to make an equation that what says is I'm going to grow if my replication rate is larger than the average, right? So if I'm faster than the average population, I will replicate. If not, I'm going to go down, okay? Then we can do actually a number of interesting things from that. For example, an exercise that you can consider is, imagine that I have this several population system, right? I can tell you, easy to do from computer simulation, that if I plot the behavior of this system, what I'm going to see is that starting from the same initial value, imagine, for example, that every population is exactly the same, right? So xk at time 0 is 1 over s, so that way it's normalized. Some will grow, some will decay. Some will decay immediately, some might go up and down, some will go into the maximum population, right? But this sum is only one species. So how do you prove, actually, that under these conditions, there's a single winner? The fittest, in a way, under this simple formulation, okay? So that's what defines the basic model I can construct. And an important message from this model, and we need to think about what's missing here, is that the only outcome is that it's one species who wins, the fastest replicator, right? The fittest, in a way. So what will be needed to avoid that? Because in nature, for example, we see populations that compete for resources, but they do not exclude each other. So diversity is maintained, right? So we need to think how we will expand this theory to allow that to happen. So that's for competition. How can I take into account something that also happens in natural system, which is cooperation? That I am not just trying to overcome other partners in my ecosystem. I want to help them. So I want to enter into a situation where we have a symbiotic relationship, and in a way or another, we help each other. And what are the consequences of that, and what's important? That's the second part of the story, cooperation. One of the reasons why we have been such a successful species in evolution is that we were amazing cooperators, right? We tend to think in humans also as the one instance of the survival of the strongest, in a way. But cooperation was crucial for us to succeed. And cooperation is a process that is there in nature in many places and pervades the emergence of big transitions in evolution, like for example when single species started to get together, right? Because the cooperation in a way or another was relevant to achieve new levels of successful evolutionary complexity. So how can we make a model of that, and what kind of case studies can we consider, right? Well, cooperation can be there in a broad number of ways. For example, things that we observe or that we can engineer, right, is that a given species, for example, of a bacterium, creates a metabolite, right, which is needed for another species in an obligate way to replicate and thrive, okay? But the other way around also happens, that I do create a metabolite that this species needs necessarily to survive. If that's the case, we have this kind of loop, a cooperative loop, where both species need each other, okay? It has been conjectured, and that's a very important idea in terms of the transition to living systems, that happened also in an early biosphere when RNA molecules, which are a potential candidate for the origins of life, were able not only to code, to store information in a chain, but because they can fold, they could be molecules with some enzymatic features. And the conjecture that we have, and it's partially been shown in the lab, is that if you can fold and be an enzyme that catalyzes the replication for another RNA, and the other way around, this catalytic system, right, that has been called a hypercycle, has the capacity, because you have a cooperation loop, right, to make this system to thrive, right, and actually go through some information thresholds. We'll talk about that in some other lecture about quasi-species, but that was a crucial concept that also seems to be present in ecosystems, right? Species that, in a way, act improving, improving the context of a given species that also helps the first one to grow, right? That could be, for example, for earthworms in the soil. They improve the quality of the soil, plants thrive, and the plants contribute to improve the quality of the soil by storing carbon, and we have, again, an important loop. An important consequence of this that we'll discuss in another lecture is this concept created by Lynn Margulis, that symbiotic loops have been really essential to actually allow complexity to emerge in the plant. In terms of bioengineering, we'll also see that this is a very important thing because we can engineer this kind of metabolic or replicating loops, and that has a lot of consequences. Now, how can I make a mathematical model of this, and what's the consequence of this? It's easy from what we just saw in the competition model, because essentially what we are drawing here is a system where, again, I have two species with given populations, okay? Each one has to replicate, all right? And it will at some rates, but, and let me use some color to make a distinction here. To do that, each one needs the other. So to make this clear, I need you to replicate myself, and you need me to replicate yourself. So I have a closed loop where R1 and R2 are not just parameters, are conditions that require the presence of the other population. How can I make a model of that? It's easy, because actually, if you think about how can I write down what I just summarized here, that's going to be our formal description of this hypercycle. What I just need to do is the following. Have in mind that interactions are needed. X1, X2, they have to interact as directly, like here, or indirectly, like here, okay? So I'm going to replicate. That's what we just wrote down for the competition system, but now, unless the other species is there, and interacts with me, those terms are meaningless. So I need you interacting with me. Remember that we defined that as part of the mathematics of interactions. You have the product of the two populations, and you need me. So that's the simplest way we can introduce the idea of cooperation in terms of the mathematics that we need. Now, we keep in mind that we keep assuming that we have a maximum size of the total population here, and we keep using the constant population constraint, right? In this case, as before, I'm assuming that the fractions of populations for each compartment, for each species if you want, is normalized, is 1, right? Again, this allows me to simplify the system. So we'll have the phi functions, but the construction, the mathematics I need, is exactly the same. There's no difference from what I just used for the competition system. Under this constraint, these phi here, right, in this case, are not just R1, X1, R2, X2. They have to take into account everything here and everything here. But it's easy to show that this means, and that's going to be something very common here, that I have to consider everything that is here. All the kinetic terms that actually define how things interact or grow or die, right? So it's 2 times the same thing. No, sorry, it's this here. And to make things simpler and to write down a nice result where you can consider the generalized problem, let me use this particular case, the symmetric case, which allows to write down easily the equation for the competition process. And if I do that, right, this population here, for example, I just choose one of them, right, can be written, the differential equation, as this. R1, X1, X2 is just 1 minus X1. Okay, so it's easy. Okay, I have this term, now I have minus X1 multiplied by my phi function, which introduces the actual way we compete with each other, right? So this is just this here, right? Except that R1, R2 is just the same thing. So it will be 2 times R X1 X2, okay? Just aggregating terms. No, sorry, I made a mistake. This is X1 X2. That's correct. But again, X2 is just 1 minus X1. You can see that I already have a single dimensional equation, one species equation. And this can be written as follows. We have R1 is just R, right? I have R here repeated. So if I aggregate terms, I have all this term here is already here too, okay? So this can be actually, since I have a 2x1, written in this way. Quite nice. Everything collapses, is factorized. Let me make a small change because it's going to be helpful to interpret. I can put these 2 here, multiplying, and this means that I have this here. This is my equation for the cooperation system. All right? Reminds us a little bit what we just derived, but it's an extra term, right? That could be kind of a logistic kind of behavior, but there's something else here, which is quite different. First thing, one thing is easy to do is find out who are the fixed points, right? The potential equilibrium points. 0, 1, and the extra one, 1/2. These are my 3 potential equilibrium points. Now you can use stability analysis, as we already show how to use, right? You take this system, you find the lambda, you see the sign for each point. It's easy to show that these 2 points are unstable. You are not going to go into 0 or 1 unless you are already there. They are going to be unstable. And the point that defines that you are just the average population, you are just half of it, is stable, okay? Which is very interesting because it means that the behavior of the system will tend to actually go into half and half of each population. That the result of the dynamics is nothing but flowing into a solution that would always be share the system, okay? If I think in terms of the dynamics, okay, what I'm going to see is that no matter where I start, right, if I start a little below 1, say x1, right, this will be 1, I go down into 1/2, no matter where I start, okay? So 1/2 is an attractor of the dynamics. And I will have a similar drawing for x2, okay, for sure. So how the potential function of this system looks like, right? That's an exercise for you. This is a cubic system, have 3 times x. So the potential function, if you remember, is defined in this way, where f is just the function that describes how the dynamics goes. This is a cubic polynomial. So if you solve that, you'll end up into a, I don't have it written here, I can give you a solution, but you'll have a 4th order polynomial, right? And if you draw it, that's part of the exercise, right? What you will have is the following. Is a function that goes like this, right? This is my potential function. This is x. Remember that the potential function captures also the nature of the equilibrium points. Here, and I write down as with an open circle, and here an open circle. This is 1, this is 0. I have my marbles that will fall into what? Into the only attractor and equilibrium, stable equilibrium point. 1/2. Here and here, right? Remember that by construction I write this as a mathematical function, but this is forbidden. This is negative populations, and this too, because this will be something that I do not allow to happen. I cannot have more than 1. This is the maximum, right? But you see the shape. What would be just to have the comparison, the shape of the potential function for the competition problem that we have discussed before, right? That's also an exercise for you. For the problem we just solved, the potential function depends on, remember, the balance between replication rates of each species, okay? If you write down this, you calculate and plot it, right, for the problem I just defined. That would be cancer cells, just by maintaining what I was saying before. There will be a potential for the cancer cells. This will be stable, so 0. And this will be unstable, right? So this will be the solution for extinction of cancer cells, right? if my replication rate is smaller than the normal tissue. I cannot overcompete normal cells. And you will find the reverse of this picture. Let me draw it here, right? I have maximum here, minimum here. Okay, sorry, this was cancer. Where this would be unstable, right? The zero population for cancer, whereas this will be stable. It will roll down into overcompeting normal cells. Okay, you can see that by comparison that we have very different potential functions. Okay. So when is each part going to be relevant? Well, you see, if I want to maintain diversity in a system, cooperation is a good mechanism because you help each other. And by necessity, we either go extinct, the both of us, right? That's in a very general scenario, not in the way we have defined. Or in the case of cooperation, right? You help each other, so you get into a balance of values. If you've solved this under this more general picture, what you will find out, and that's quite intuitive, is that just the location of these moves, right? But the essential picture is exactly the same. So where can we apply that? As we will see in different lectures in many instances. Let me just finish with some thought that I think is interesting to consider as a case study, right? Which is an idea that was formulated many years ago, or some years ago, by physicist Paul Davies. A very interesting idea. He called that the Shadow Biosphere. Right? The context is the following. We'll search for life in other planets in the universe, but what if something happened at the beginning of the development of life in our planet, which might have emerged in a number of times, not necessarily once, and what could have happened if some different kind of life, maybe even not based in DNA, is in our planet, and we just fail to detect that kind of life. That's a very interesting possibility, and I want you to think about what are arguments against and pro this coexistence of our life and the life we haven't found, the life in the shadow biosphere. Could you formulate? And what kind of help can you have from mathematical models like this? Because and that's my conjecture, if there was a shadow biosphere, it would necessarily compete from basic resources with the very successful biosphere that we know, right? Is this necessarily meaning that the two major classes of life have been competing and one won over the other? And if not, why not? What part of the mathematical model you could formulate using competition might fail to capture that possibility? And maybe there's a shadow biosphere somewhere around.